when a patient has blood drawn and tested, he or she can rest assured that their results will be the same no matter which lab performs the tests. The rates of inter-laboratory agreement in blood testing are greater than 99%, but not so in immunohistochemistry for analysis of cancer biopsies. Different labs can produce different results on the same patient sample. In this published example, you can see serial sections of a breast carcinoma as it appears under the microscope. The sections were stained for estrogen receptor at 2 different hospital laboratories as part of a national proficiency testing survey. As you can see, Lab A shows lots of positive tumor cells stained brown, whereas Lab B shows none. Different test results on the same patient sample that lead to different cancer treatments. Published studies show that discrepant immunohistochemistry test results happen around 10 to 30% of the time. A cancer patient's treatment shouldn't depend on which lab performs the test. So synchronizing immunohistochemistry testing around the world requires the creation of standardized samples known as reference standards and calibrators. Here are some examples of such reference materials for the clinical chemistry laboratory. Using these kinds of reference materials, inter-laboratory reproducibility in blood testing is greater than 99%.
There are no analogous reference materials for immunohistochemistry testing. With support from the National Cancer Institute, my colleagues and I here at Boston Cell Standards solve this problem. Here's how. Immunohistochemistry is used to detect one type of macromolecule amidst thousands on a cell. This animation illustrates diverse proteins on the cell membrane, with one of them being the assay target. So, how to create an immunohistochemistry reference standard for that molecule? The cells themselves aren't suitable reference materials. Boston Cell Standards developed a synthetic cell equivalent with defined molecular concentrations as needed for a calibrator. We first attach a fluorochrome to the purified macromolecule. There's no measurement standard for the many different cellular proteins, but there is one for fluorochromes. We then attach the purified molecules to cell-sized microbeads, such as are used to standardize flow cytometry measurements. For every calibrator, we calculate molecular concentrations on the microbead against a fluorescence standard at the National Institute of Standards and Technology. One NIST standard serves for all immunohistochemical assays. These microbeads are then fixed in formalin and suspended in a clear proprietary liquid. When pipetted onto a slide, the liquid suspension hardens, keeping the microbeads on the slide during all subsequent steps. This image shows stained cells and the microbeads side by side. Some microbeads are unstained because they're negative controls. This system works for any immunohistochemistry biomarker. These are the first and only immunohistochemistry calibrators. They provide exact biomarker concentrations for aligning IHC testing worldwide. And they can be mass-produced, delivering to every lab the exact same standard. Bottom line, the introduction of the first IHC reference standards supports optimal patient treatment regardless of where the patient lives, regardless of where he or she is treated. It ensures a consistent application of the best that medicine can offer. It's a win for pathologists who have greater certainty that the results are accurate. It's also a win for oncologists who will have greater confidence in patient diagnosis and treatment selection. And, of course, it's a win for cancer patients.